Hello and welcome everyone to another of our i5k webinar series. It looks like there's a lot of new participants this time, and I hope that you'll continue to join us in future webinars or take a look at the other webinars that we already have up on our YouTube and our website. So just a few things before we get started. We are recording this webinar and we'll be posting it, like I said, to our YouTube channel, and the slide deck will be on the website. So please look there for more information. We want to have an engaging discussion with you after the presentation, so please submit your questions to us in writing. And you can do this one of two ways. One is to locate my name at the top of the participants list. My name is Anna Childers, on the side panel, and you can either single or double click that depending on if you have a PC or a Mac, and that's going to open a private message window through which you can send your questions. The other way that you can do it is that you can send an email to me at my email address, and I will send that to everybody right now through the private messaging function, so you should have that. And so again, send those please just to me so it doesn't show up on everyone's screen, and as you can see, that will knock our that will knock our presenter out of the presentation, so we don't want to do that. Now, finally, for those of you that are calling in as a group, if you could send me a message or an email and let me know how many participants you have joining us from your location, that would really help us keep track of our numbers and how well we're getting our seminars advertised and how much interest we've got. So finally, with that, I want to introduce our speaker, Dr. Keith Hopper from the USDA-ARS Beneficial Insects Unit. And Keith, I'll let you take it away and inform everybody about your webinar. Thank you.
Well, thank you, Anna, and thank the i5k group for inviting me to talk about this subject that I've been working on for a long time, which is about the host specificity and evolution of host specificity in parasitoids being considered for biocontrol introductions. As you can see from the first slide here, there are a lot of people involved who contributed to this, and from a number of institutes, with funding from my agency, ARS, but also from AFRI, NSF, and the North Central Soybean Research Program. Oops. So, what I'm going to be talking about are parasitoids of aphids in the genus Aphelinus. There's something like 90 described species, although we've just described 5 more species in the last few years. There may be, 10 times that number when we're done. They have short development times. They develop from egg to adult in about 3 weeks at 20 degrees. They're synovogenic. They produce eggs throughout their lives. And they host feed on their hosts to get nutrients for egg production. They don't carry many eggs at a time, 8 to 30 eggs, but they can parasitize hundreds of hosts during their lifetimes. They're long-lived, at least in the laboratory, living 2 to 4 weeks. They're very small, as you can see from the image here. Let's see. This is a wasp. It's about the size of a printed period at the end of a sentence, 1 millimeter long or so. And they are weak flyers. They basically just hop rather than fly. And they, like most Hymenoptera, they have haplodiploid sex determination. So during projects to find parasitoids to introduce against Russian wheat aphid and soybean aphid, we've explored throughout Eurasia and found parasitoids in 3 species complexes: the Avaripes complex, the Malai complex, and the Ascis complex, all the way from Morocco over to Japan. And this material is what I'm going to be talking about. During exploration, we brought material back and tested host specificity in the laboratory. We did this with no-choice experiments where we put 100 aphids of mixed stages of a single species on plants in cages, and we added a single female wasp to each cage, left her for 24 hours, and then recovered her and measured the number of aphids parasitized, the numbers of adult progeny that emerged, and the sex ratio. So what we were measuring is a combination of host acceptance and host suitability. We also did behavioral bioassays on specificity, where we directly observed females with aphids. In this case, in a small arena, about centimeter across and 50 millimeters deep, where we had aphids on the excised host plant leaf, again, a single wasp, and we observed them directly for 25 minutes. And we measured things like the numbers of aphids approached, stung, oviposited in, mummified, as well as looking at behavioral defenses. So I'm going to talk about several designs that we did. For measurement of specificity, we used 17 aphid species in 8 genera on 11 host plant species in 9 families, and we looked at many parasitoid populations. So for these experiments, we measured something over 3,000 wasps. And then for the behavioral host specificity, we only looked at 4 parasitoid species. Ones that passed the first round of testing had narrow host ranges, and we wanted to know why they had narrow host ranges. So we observed their behavior, dissected half the aphids to see if they laid eggs, and reared the other half of the aphids to see how well the progeny survived. And then mostly what I'm going to be talking about today is experiments to look at the genetic basis of host specificity, where we did an interspecies backcross. Turns out we can hybridize Aphelinus certus, which doesn't parasitize D. noxia, with Aphelinus atriplicis, which was collected from D. noxia and does very well on it. We backcrossed the progeny. We backcrossed the F1 females to certus and looked at the phenotypes and genotypes of 320 backcross progeny. And we measured specificity, as I described previously in these cage experiments where we left the females for 24 hours. And this is the sort of results we got. On the y-axis is the numbers of mummified aphids in a 24-hour exposure. On the x-axis are 7 aphid species that we tested in this first round of testing. And this is the box plot where the boxes are the solid lines here are the medians, and the edges of the boxes are the 25 and 75% quantiles, and the dots are outliers. The diamonds are means, and the error bars are 95% confidence intervals. Many of the parasitoid species had broad host ranges, and so we didn't pursue them further. However, we had several species, Aphelinus hortii, for example, which is a specialist on D. and then Aphelinus glycinus and Aphelinus rhamni, which are specialists on soybean aphid and its relatives. I want you to look at, in particular, though, at atriplicis, which was collected from Russian wheat aphid, D. noxia, and parasitizes it very well, but it has a very broad host range. And Aphelinus Aphelinus certus, which I'm here, which doesn't parasitize D. noxia at all. Turns out these are allopatric in nature, but they will hybridize in the laboratory. And so we took the advantage of that hybridization to look at the genes and that allow atriplicis to parasitize D. noxia, but prevent certus from parasitizing it. So to back up a second, we wanted to look at the genetic basis, and one of the things that we did was we sequenced and assembled the genomes and transcriptomes of certus and atriplicis to allow us to look for genetic differences. We also did this with 14 other species of Aphelinus, but I'll only be talking about atriplicis and certus today. So the pipeline for doing this, we extracted DNA from a single haploid male to avoid problems with heterozygosity and structural variants. And you can do a library from a single haploid male, and we made libraries from these and sequenced them on Illumina HiSeq using 100 to 150 nucleotide paired-end sequencing to get about 15 to 400 gigabases per genome, which gave us 100x coverage. It's a mute mic.
Really, what would be really interesting, and from the context of what we just talked about.
Somebody has got their mic unmuted, so I'm hearing them talk.
We fixed it.
Did you mute your mic? Okay, thank you. Assemble these genomes with using the CLC Bio Genomics Workbench using De Bruin graph, which uses De Bruin graphs, and we got various quality statistics, including M50s, numbers of contigs, and also we checked what proportion of the busco arthropod genes we found in our assembly. And then we've annotated these genomes using Augustus for gene finding with the Nesonia vitripennis models, and then blasting those genes against the protein sequences, rather, against NTBI-RefSeq to find homologs, and then using blasted geo, that is gene ontology, to get gene ontologies. And because a fair number of genes didn't have any hits on GenBank, we also used InterProScan, which uses a lot of hidden Markov models to look for domains that could give us some idea of gene function. And right now, I'm running OrthoDB with all of the species we've done to find ortholog and paralog clusters among Aphelinus species. And I'll get back to that later. We also did transcriptomes for most of these species that we've done genomes for, and we mapped the RNA-seq data back to the genome assembly to confirm that the genes that we were looking at, that we found with Augustus, were actually being expressed, and to look at expression differences among between atriplicis and certus. I'm not going to go through, this is a big table, I'm not going to go through all the details of it. As you can see, we've done many species, sequenced and assembled their genomes and done annotations, and a paper will be coming out on this next summer. What I want to show in particular is that here's atriplicis and certus, which are rather similar in genome size as estimated by flow cytometry. The assembly sizes are smaller than the sizes estimated from flow cytometry, and the assemblies are rather balkanized. The numbers of contigs are quite large, like around 35 to 40,000 contigs, because of presumably of repetitive DNA. Nonetheless, we get busco scores of like 93%, over 90% of the busco genes are present in our assemblies. And we end up with about 25,000 genes in each species. About half of them single copy, and the others varying in copy number from 2 to 300 copies, or paralogs. Among these, we found about 100, over 90 chemoreceptor proteins. But in Nesonia, people have found like 500 chemoreceptor proteins. So we probably very underrepresent in our annotations the numbers of chemoreceptor proteins, probably because they've evolved rather rapidly. We also found a fair number of cytochrome P450s, which could be involved in using hosts on nasty host plants, and a fair number of venom proteins. Over 90% of the genes did have transcripts from the RNA-seq data. About 80% of the genes had blast hits, and of those genes that have, of the total number of genes, only about less than 40% had functional annotations in gene ontology. So that becomes important when we start looking at the genes underlying host specificity. We used this data set, these the proteins that we've discovered in this complex, to generate maximum likelihood phylogeny, molecular phylogeny, which I show here. And the idea is that we wanted to see whether host use was phylogenetically conserved. Were species of wasps that were closely related more similar in host use than species of wasps that were distantly related? So we generated this phylogeny, and then we mapped host specificity as shown in the figure on our behavioral bioassays, or sorry, our 24-hour bioassays, where we looked at the numbers of aphids parasitized of different species, we map those onto the molecular phylogeny with the idea that if host use was phylogenetically conserved, closely related species would show the same level of parasitism of an aphid species. However, if host use was associated with speciation, with cladogenesis, we would expect closely related species to show very different host use. And the answer is both. So unfortunately, it doesn't look like there's a consistent pattern in the phylogenetic conservation of host use. For aphis glycines, closely related species show the same levels of host use. By the way, this is from 0 to 100% of the maximum number of hosts parasitized. On the other hand, if you look at Diarrhea noxia, closely related species are divergent in host use. So Hortii and Kurjamavi doesn't use Diarrhea noxia. Hortii is a specialist on it. Same thing with atriplicis versus certus. Atriplicis parasitizes D. noxia, certus does not. So, to further understand what the genetic basis is of the differences in host specificity that we're seeing, we took advantage of the fact that we could hybridize atriplicis and certus and in the laboratory, and certus does not parasitize D. noxia, atriplicis does. So we took female certus and crossed them with male atriplicis to produce an F1 hybrid. And this is just a little thing. This is the P is the gene involved in being able to parasitize D. noxia, and these are nucleotides flanking SNPs flanking the gene. And then we back-crossed the F1 hybrid to certus, and obtained back-cross progeny, some of which were recombinant, some of which weren't. They would either be heterozygous for the gene involving parasitizing D. noxia, or homozygous not parasitizing D. noxia. So to map the quantitative trait loci involved, we used a pipeline that Randy Weiser and I have developed at the University of Delaware, which we've called RASP, Restriction Site Associated Sequence Polymorphisms. It's a variety of RADseq, where we restrict with restriction enzymes, ligate on adapters that are barcoded, and then we can multiplex these. We do a step where we size select, amplify to add more adapter sequence, size select again, and then sequence on Illumina HiSeq, usually just doing 100 nucleotide single-end sequencing. Then with Sean Polson at DVI, we developed a pipeline for analyzing these data that uses actually existing software for mapping and for quality control, mapping and variant calling. We call this pipeline REDREP, for reduced representation library analysis. It's available on GitHub if you want to get the code, and involves three main steps: a quality control step that involves sequence trimming and barcode deconvolution, and mapping the sequences to a reference, mapping the reads to a reference, and then calling variants. And the quality control step involves a FastX toolkit and a CutAdapt program. And then we use BWA MEM for mapping reads to the reference. Or you could also do a de novo clustering if you don't have a reference to map to. And then we use we were using the Unified Genotyper in the GATK toolkit, and now we're switching to their haplotyper. But we use that for calling SNPs. And then once you got your SNP set, you can use other programs to analyze the data. This pipeline, the lab part of the pipeline, costs $10 per sample for library prep, and we can multiplex up to 394 samples per lane of high-seq sequencing, and the sequencing ends up costing, at that rate, would cost about $4 per sample. So it's relatively inexpensive. One of the things that Randy and Heather found was published in this manuscript that's in prep, or in review rather, is that even if you have a good reference, like with maize, if you're just looking at SNPs, you can confound paralogs so that you get pileups of more than 2 haplotypes, because copy number variants are confounded because the reference genome isn't the genome that you got your reads from. So if you look here on this figure, you can see this is a number of different copy numbers, and what you would expect are these gray bars, which are 2 copy variants, because this is a cross of 2 inbred lines. But if you get more than 2 variants, that's actually not possible, or extremely unlikely given the inbreeding on these lines. And what we think is happening is it's a pileup of haplotypes. And this was found by using a locap program developed by folks at I think University of Chicago, which we've incorporated into our pipeline as well, which allows you to scan across a read and develop a haplotype for that read, so that if you have more than one SNP in a read, you can count the number of types of variants that you have. For example, here you have 1, 2, 3 haplotypes in this subwindow. And as I said before, you can't. That's physically impossible with a cross between inbred lines. And so this results from piling up paralogs without knowing you're doing it. So if you looked at SNPs, you would just call each of those as a valid SNP, but you wouldn't realize that you're actually piling up paralogs. Anyway, so I took the data from the genotyping and used RQTL to develop linkage maps and to analyze for QTL. And these are the LOD scores, these are the chromosomes, and these are the QTL we found. We found 8 QTL, as you can see here on this linkage map, on 3 of the 4 chromosomes. By the way, these are the karyotypes of Aphelinus atriplicis. There were actually large numbers of markers, but thousands that you can genotype this way. But since I only had a mapping population of 320 individuals, I didn't use all those markers because it would be overkill. I ended up using 274 markers, which are about 2 centimorgans apart. And this, the linkage map size is 654 centimorgans, and given the genome size of 350 megabases, that's about half a megabase per centimorgan. Anyway, so there's 8 QTL, and these QTL interact with one another, and the state of one QTL affects the effect of other QTL. And by the way, this paper is in review at Evolution describing these results. Anyway, the 8 QTL together, including their reactions, explain about 40% of the variance in parasitizing D. noxia. And each QTL alone has large effects, and half the QTL interact with one another, as I said before. Unfortunately, because of the relatively small mapping population, 320 individuals, the confidence intervals for the QTL, the number of centimorgans that they cover, can be quite large. Some of them, actually I can show you more illustratively here. This one, for example, looks like it probably includes multiple QTL, but I can't resolve them because I don't have sufficient recombinance. anyway, anywhere from 6 to 130 centimorgans are covered by these QTL. Nonetheless, we were interested in what genes from our genomic assemblies and annotations were under these QTL, and particularly what genes that were highly sequence divergent or highly expression divergent that fell under these QTL. So we looked at rates of non-synonymous to synonymous substitutions among our genes, and 2% of the genes had rates of non-synonymous substitutions threefold or greater. So out of 25,000 genes, that gives us about 500 genes. And about half of these have gene ontology annotations, so we have some idea of what they do. 40 are potential chemoreceptor proteins. These others, we don't know what they do because they probably evolved too rapidly to have homologs in GenBank. If we look at sequence-divergent genes expressed only in females, because we think it's females who are deciding whether or not to oviposit in hosts, and that's what's determining host specificity. If we look at sequence-divergent genes expressed only in females, we go from 500 genes to 29 genes. And unfortunately, here, there are no chemoreceptor proteins identified. And again, about half the genes don't have any annotations. So even if they have a blast hit, the hit is a putative protein with unknown function. However, analysis of the protein domains identified some of these as having transmembrane domains and signal peptide domains, which suggests that they might be chemoreceptor proteins, although about one-third of genes, one-third of proteins have transmembrane domains because many proteins are embedded in membranes, so we really don't know what these do yet. We also looked at expression divergence of genes expressed just in females and looked at expression divergence of 100-fold or greater, and again, we get about, in this case, 21 genes, some of which are potential chemoreceptor proteins, and in both cases, there are several potential transcription factor proteins that could be regulating genes involved in behavior. Now, to see if these expression and sequence divergence genes were associated with QTL, we at first tried to co-hybridize markers for the QTL, flanking markers for the QTL, and the markers for the genes, the probes from the genes, onto chromosomes. And we're able to do this in principle. So this shows you a SIM structured illumination microscopy image with chromosomes stained with BAPI, and this is the 28S ribosomal gene, which is a multi-copy gene family, which you can readily see. Unfortunately, in our hands, it's very hard to keep the chromosomes on the slide, and also with metaphase chromosomes, we can't map very precisely. So we switched to a different approach, a genetic approach, where we use Kompetitive Allele Specific PCR, CASP, which is a protocol developed by a company in the UK that uses 2 allele-specific primers that will release a fluorophore when they're amplified using RT-PCR. And I'm not going to go into all the details of the protocol. You can get the paper from me if you want. What you end up with, though, is 2 different fluors, FAM and HEX in this case, that give you different signals depending on whether you're a homozygous atriplicis, homozygous certus, or a hybrid. And so that allows us to phenotype all the individuals, that's what this is, phenotypes of the 320 individuals, and map those, genotype 320 individuals, but the genes of interest, and map those onto the same map as the QTL. When we did that, we've done, I think, 15 genes so far, and most of the ones that we've looked at that were sequence divergent and or expression divergent mapped to QTL. And a lot of them mapped to QTL 1, 2, 3, 5, 6, 7, and 8. A couple of the divergent genes don't map to QTL, but it may be that we just didn't find those QTL. And each of these genes, the state, whether it's certus or atriplicis, has an effect by itself. But the effect sizes are much smaller. Here it's about half an aphid parasitized, one aphid to half an aphid parasitized, whereas the QTL, it's like 2 to 4 aphids parasitized are the effect sizes. Anyway, now what we want to know for these genes that, by the way, so like, for example, this is a gene, was it 6995, which has a signal peptide and transmembrane domain, has an effect on the phenotype. We want to determine, using tissue-specific expression, whether that's expressed on cincilla, on antennae, or ovipositors. And so what we're doing right now, this is a scanning micrograph of the club of a female antenna, and we've developed probes for these genes, and we're hybridizing to the antenna. This is confocal image of this part of the antenna. Actually, this part of the antenna is about right there. And this is the base of a cincilla where the candidate gene is being expressed. We're going to switch to doing sectioning because this spatial resolution for just standard confocal is not precise enough to show that this is actually being expressed on the membrane of the cincilla. So we're going to switch to sectioning and doing structured illumination microscopy or storm microscopy. Lastly, I just got a $500,000 grant to pursue this research further, and particularly to develop magic recombinant inbred lines to do high-resolution mapping. So our QTL are span less than a centimorgan, and test where the sequence and expression divergent genes fall under these QTL. Use a larger mapping population as well, so we can find more QTL, and then test, like we've been doing, try to test whether these are expressed in the tissues that we think they should be expressed in. And lastly, using the transcriptomes we've developed and doing more transcriptomic analysis of individuals, we will phenotype and look at expression differences among individuals that have different host specificities. And for this research, I'm going to be looking for a postdoctoral scientist at a GS9 level and a research assistant at a GS7 level who know quantitative genetics and hopefully some bioinformatics as well. And that's about it. Any questions? I'd be glad to answer them.
Wonderful. Thank you, Keith. I just want to remind everybody that if you do have questions, go ahead and send those to me, Anna Childers, either using that private messaging function within AT&T Connect by clicking on my name, or go ahead and send an email to me. And also, just another reminder, we've had a few more people join. If you've got more than one person joining the webinar from your location, if you could send me the number, that would be very helpful. So let me get to the questions, though. I've already received one question. Chemoreceptors are notoriously difficult to predict computationally. Since it seems that you're particularly interested in those candidate genes for parasitism, are you working on manually verifying how well you predicted those?
Yes. And I mean, actually, the way we're doing it is by looking at if the genes that we think are chemoreceptors are being expressed on cincilla membranes, along with ORCO, the co-receptor for chemoreceptor genes. So we're actually trying to verify it experimentally, which I think is really the way to do things if you're trying to verify function, is to do it experimentally. And ultimately, we want to knock out gene function using CRISPR-Cas9.
Wonderful. Have you noticed, is there a sweet spot in terms of the amount of sequencing data, number of individuals per population that you're working with, as well as the number of populations that you're analyzing in order to get a good feel for this trait?
We don't know yet. I mean, for doing the assemblies, we're just trying to do the initial assembly using a single individual. And I haven't gone into the details here, but we've actually done, to get better assemblies, we've done 50x coverage with PacBio. But to do 50x coverage with PacBio, we have to grind up, thousands of WAS together. because you need on the order of 10 to 20 micrograms of DNA rather than 100 nanograms. And that ended up being, those data ended up being really, really hard to assemble because of heterozygosity and structural variants just making it really difficult. And so the problem is that PacBio may not be the ideal long-read system for insects as small as this. So that what we're working on now, in fact, we're preparing the library, we're preparing the DNA actually for 10x sequencing, which is a Lumina-based sequencing technology that involves pooling together DNA from the same region and doing sub-assemblies of it. And with that technology, you can use as little as a nanogram of DNA. And so we can use a single male wasp and do 10x libraries. So we're trying that. The other thing we're doing is we're inbreeding, doing brother-sister mating for 10 generations as part of this, this development of recombinant inbred lines. And we're going to use those inbred individuals to try doing PacBio sequencing again with things that are homozygous.
Okay. So I think
but to answer your question, sort of, we use one individual to do an assembly, and then we resequence with large numbers of individuals. So, like for mapping populations, hopefully 500 to 1,000 individuals. For these recombinant inbred lines, we're going to have 400 lines, but we're going to sequence many individuals per line to get around experimental error problems.
Okay. So that might feed well into this other question. So would your workflow need to be significantly modified if you could get, if you had even smaller species? It sounds like, though, you've maximized this for the protocol and the sequencing approach for very small insects.
Right. I mean, these are about, there are smaller insects, but they aren't much smaller. I mean, these the insects themselves weigh about 10 to 15 micrograms, and they only have about 100 nanograms of DNA. They are very, very small insects. And so I think if you had much larger insects, you could maybe, for example, do a PacBio library from a single insect, whereas we don't have that luxury.
Okay. And in terms of modifying the workflow also, would you need to modify it if you were looking more at wild populations instead of lab populations, or, populations in which there would be larger heterozygosity, possibly because you don't have a haploid male?
Right. I mean, for assembly, that's going to be really hard. You're going to have a lot of heterozygosity, and assembly is going to be hard.
Would that also?
It also affects,
I was
if you have a reference that you can map to for the reduced representation library protocol, there's a real problem with the quality of your reference. How close it is to the organisms that you're actually testing. So this is a big sort of 800-pound gorilla that's not mentioned in a lot of RADseq papers. Is that there's not a lot of sequence homology. You end up having to throw out, I've done this for wild populations where I was doing phylogenomics, and you have to throw out like 80 to 90% of your data if you're trying to do phylogenetics, because there isn't, for anonymous loci, which you get when you're doing RADseq protocols, there's very little sequence conservation, particularly between species. But even within species, there's lots of variants that just don't occur across all individuals. And so it's a problem if you don't have a good reference. The same thing is true with Cas genotyping. We actually sort of struck out with it initially because we were using a reference genome for doing our genotyping, for determining the sequence for the genotypes. And it turns out you really need to use the parents or grandparents of the lines that you crossed if you want to develop primers that are going to work in the individuals in your cross. So if you're working with wild-type populations, it's really problematic.
Okay, and that would that ploidy issue and everything also come into play in terms of being able to call SNPs? I know you mentioned that paralogs are a problem, but if you've got if you can't sequence a haploid male, is that also going to complicate? that situation?
Well, for the Cas genotyping, we don't care whether they're heterozygous or homozygous, but it would be a problem if you had paralogs, if you weren't looking at a single copy gene. If you're confounding paralogs, then you're going to screw up the inheritance completely. So you really do have to have some way of knowing that the things that you're looking at are orthologs.
Okay, wonderful.
That is, it's the same gene in the same position.
Okay. In terms of the assemblies, do you think, you mentioned scaffolding, would that have been a benefit to your research if you had a really good reference assembly for one of your species?
Well, it's interesting you asked that, because that's exactly the reason that I took one species, atriplicis, and actually did mate pair and PacBio libraries and used them for scaffolding, and did a sort of a homebrew scaffolding thing where I used the Genome Workbench to do the initial assembly, and then use S-space with and PB jelly to scaffold. And that increased the contig sizes like 5-fold, but interestingly enough, it hardly had any effect on the numbers of genes I called or the BUSCO scores. I added a couple of percentage points. I went from like 92% of the BUSCO genes. So this is a set of genes of like 2,700 genes that are found in 90% of arthropods, and I find almost all of them. And if I do a 5-fold better assembly is measured by the N50, I maybe increase the number of genes I'm finding by 2%. So how good an assembly you need depends on the question you're asking. But it also depends on the species, because I've done the same thing, the same sorts of things with Heliothis fluorescens and Heliothis simplexa, and those assemblies are terrible. You get, BUSCO scores of 50% of the genes, and the genomes are very, very fragmented. I think it's just that Lepidoptera have a lot more repetitive DNA, a lot more relic transposons than are found in the wasps that I'm working on.
What would you say is the largest either interpretation or informatics challenge in your workflow?
Gene annotation, figuring out what the genes do. That's the hardest thing is figuring out what the genes do. And especially because the genes that are of most interest to us, the things that are involved in local adaptation, appear to be evolving really rapidly, and therefore don't have a lot of homologs. And so finding function from homology is difficult.
So do you have a plan for working around that?
Just like I said, looking at tissue-specific expression. So if we think that the genes are candidate genes are chemoreceptors, well, see if they are being expressed on sensilla, on antennae or ovipositors, and if they're co-expressed with the olfactory receptor co-receptor gene, and then doing knockouts. I mean, I'm still struggling with doing using various tools to look at protein domains, but that quickly runs into barriers of there's just not enough information out there for there's not enough sequence conservation for things like chemoreceptor genes to be able to infer function from homology.
Okay. When you're doing, you're trying to infer gene descriptions and information, what cutoffs are you using in order to make those inferences for divergent genes using InterProScan or Blastigo?
You mean what for hits to like RefSeq?
Yeah.
I use one in E value of 10 to the minus 3, just as for RefSeq, for blasting things against RefSeq. Is that the sort of thing you're asking?
I think that was what they were looking for.
Yeah.
When you're using your BUSCO scores to analyze how good your assemblies are you looking at the number of BUSCOs that are present, or are you looking at the number that are complete?
The number that are present single copy and by their criteria complete. I can't remember exactly their formula for calculating it. It's you know, they don't completely match the genes, the model genes, but they're complete according to the BUSCO or the OrthoDB BUSCO algorithm.
Okay. I think we have a couple more. Did the quality measures you use serve as good indicators for the usefulness of the genome assembly for your intended purposes? So I think you mentioned N50, number of contigs, and the BUSCO arthropoda scores.
Right. I didn't. There were other things that I used. The size of the genes that I found relative. So the numbers of genes, the size of the genes found relative to the genes in RefSeq, although that can be problematic because the genes in RefSeq can often be poorly annotated. Yeah, I mean, the proof of the pudding is that we're finding sequence and expression divergent genes that fall under QTL that we've mapped genetically and appear to be associated with differences in phenotype. So I mean, it's sort of like, I can't remember who said this, but intersection of lies gets you to the truth. So we're using the genetic mapping, the genome analysis, and functional localization experimentally, and ultimately gene knockout together to determine whether the genes that we're looking at are really the ones that are causing the traits. And I think you need to be doing all of those things. Just a measure of the quality of the gene assembly doesn't tell you where you've gotten there. But it looks like these assemblies are good enough. I'm still working on getting a better assembly and seeing if it makes any difference in the calling the genes or getting better annotations.
Great. Okay, I think this might be one of the more important questions here. How should people contact you if they're interested in one of your open job positions?
They should just email me. It's the best email address is my university address. The ARS server, sometimes things don't get through. They get thrown into junk. Anyways, khopper, Khopper, @udel.edu. Or if you just Google my name, you'll find the ARS website that has my information and contact information and such. And I'll add it to this slide set, too. This slide right here, I'll add my email address.
All right, wonderful. We'll try to get that. We'll have that up on the website as soon as you get to us then. And I think with that, unless there's any other questions, we will sign off for today. I want to thank everybody that listened in. I definitely want to thank you, Dr. Hopper, for helping us understand this approach. This is a big, obviously you've done a lot of work coming up with all these workflows and pipelines. I appreciate you sharing them with us. Next month, on May 3rd, we're going to have Dr. Terrence Murphy, who's a staff scientist at NCBI, talk to us about the ins and outs of genome assembly, submission, and annotation in NCBI. So if you have any questions about that, please come armed with that. And with that, I'll thank you again, and everyone, I hope you have a wonderful day. Take care.
Thank you. Bye.